In this video, we will discuss about the PARP-14 studies done in the in vivo or mice model and the signaling pathway that it will impact. And from the heading, it is clear that it will impact NF-kappa beta pathway. So we will start with this in vivo studies of PARP-14 knockdown. The PARP-14 knockdown cells are subcutaneously injected in the nude mice model, and then they check the various parameters of the tumors at different time intervals. And in the panel A, here we can see that where there is no subcutaneous injections, we can see the tumor volume size is higher compared to the other cases. In the second case, we can see when the PARP-14 silencing RNase is used, they can see a decrease in the tumor volume. And when they use only the gemcitabine, in that case also they can see the decrease in the tumor volume, and these decrease are in comparisons with the negative control. But however, when they use both the silencing PARP and gemcitabine treatment, they see a significant decrease in the tumor weight. And when these tumor volumes, when this is quantified in terms of the tumor volume, we can see that compared to the controls, the tumor volume decrease is highest when they use this silencing RNA against PARP-14 and gemcitabine. And for this treatment with either gemcitabine or PARP-14, they also see the significant decrease in the tumor volume compared to the negative control. And in terms of the tumor weight, also they find the similar results, and the minimum tumor weights are found when both the silencing RNAs against PARP-14 and gemcitabine used. And in order to validate the PARP-14 levels, they did the quantitative PCR analysis and of the mice models, different mice models, and they can see that the PARP-14's relative expression is low compared to the negative control through this quantitative PCR and also through the Western blot. So all this indicates that the PARP-14, when it's combined with the gemcitabine, it will give or leads to the lowest tumor volume. And this is in line with our previous finding that PARP-14 will make the cells sensitive to our gemcitabine. Thereafter, they wanted to check the signaling pathways that get affected. So they check major cancer signaling pathways with control and without, in control and in silencing RNAs against PARP-14. And what they are able to see is that the NF-kappa beta pathways are the major signaling pathways that got dysregulated. And these things they have further validated in PXPC3 cell lines and CPF1 cell lines using the Western blot. And here they can see that the phosphorylation of IKKB and phosphorylation of IK beta is low when they use silencing PARP-14 RNAs. And these things are true for both the cell lines, that is BXPC3 and CPF1. And this panel B is also about this CPF-CA1, where they have checked different signaling pathways. In this case also NF-kappa beta pathways are the one that got dysregulated maximum, or that got significantly dysregulated. And after that they check the P65 levels, and they can see that P65 levels in the nucleus has gone down in both the cases, and they have gone up in the cytoplasm of both PXPC3 and CFP-PAC1. And these things they have quantified in the right hand side of the panel C and panel D. Or the panel C, they have discussed about this phosphorylations of the IKKB is lower in the case where silencing RNAs against PARP-14 used. And this trend is also true for phosphorylation of IK beta alpha. And this is results are shown here. The first graph is shown here, or the middle graph of the panel C is for the BXPC3, and the right side of the panel C is for CFP-AC1. and similar trends. So similar trends they have in the bottom cases. What they have shown here the results of the P65. So P65 translocations into the nucleus goes down when the PX when this PRP14 has been knocked down, and same things they have seen that in the for this CFP AC1 cells, but in the case cytoplasm they can see the high amount of um, P65 both for the BXPC3 and SIPARP14. So all this indicates that is the PRP14 in the helps the cancer cells via nuclear translocation of P65 and phosphorylation of IKK beta and IK beta alpha. And these things are suppressed when PRP14 has been knocked down. And so finally, what this what both of these figure indicates that the PRP14 knockdown and gemcitabine has a maximum impact on the tumor volume, and it's the NF-kappa beta pathway that got dysregulated when they are treated with when cells are devoid or lack of this PRP14. I hope you understood the main findings of both of these figures. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.